Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment right now to like, subscribe, and share this analysis with your friends and family. Your support really means the world to the Sans Science Experience and helps us keep bringing this fascinating research to you. Today, we're undertaking a deep dive into the biological secrets of, well, one of those enigmatic creatures in the world of aging research, the naked mole rat, or Heterocephalus glaber. Now, if you've never heard of them, picture a sort of wrinkly subterranean sausage, and then picture that sausage living for almost 4 decades. It's incredible.
It really is. These rodents have a maximum lifespan of around 37 years.
37 years. And just to put that into perspective for you, a mouse of a similar size is ancient by age 3.
Exactly. The naked mole rat fundamentally defies the usual rules of rodent aging.
So the big question we're asking today is simple. What unique biological mechanism allows them to, well, cheat death for so long? What's their secret?
Well, that extreme longevity almost always comes back to a story about DNA. One of the central pillars or hallmarks of aging is genomic instability, basically, the accumulation of damage to our DNA over time.
Yeah.
The naked mole rat doesn't just age slowly. It's thought to have evolved profoundly superior mechanisms for maintaining its genome, especially when dealing with really catastrophic damage like DNA double-strand breaks or DSBs.
Right, those are the really bad ones, literal cuts across both strands of the DNA helix.
Precisely, and keeping those repaired is key.
And this research we're diving into targets one specific and, frankly, highly surprising molecular difference. Our mission here is to explore how the naked mole rat seems to have fundamentally reversed the function of a key protein called cGAS.
Yeah, transforming it from something that can actually accelerate aging into a massive asset for genome stability. It's quite a turnaround.
Okay, let's unpack this.
So to really appreciate this reversal, we first need to understand the human problem, so to speak. When those double-strand breaks happen, the cell activates pathways to fix them.
The repair crews.
Exactly. And the gold standard repair pathway is called homologous recombination, or HR repair. HR is vital because it's highly accurate. It uses the sister chromatid, the identical copy, as a template to perfectly restore the DNA sequence.
Which is crucial for keeping the genome stable long-term.
Absolutely crucial. Now, enter cyclic GMP-AMP synthase, or cGAS. Most listeners probably know cGAS as an immune sensor.
Right, it sounds the alarm when DNA shows up where it shouldn't, like outside the nucleus, signaling infection or damage.
That's its day job, triggering the innate immune response. But, and here's the critical paradox, when cGAS is inside the nucleus of human or mouse cells, it acts as a fierce negative regulator.
Negative, meaning it interferes.
It actively suppresses that vital HR repair pathway we just talked about. So ironically, it promotes genomic instability.
Wait, hold on. Why would our bodies evolve a mechanism where a key immune sensor actively gets in the way of the best, most reliable method we have to fix catastrophic DNA damage? That sounds counterintuitive. Kind of crazy, actually.
It is puzzling, definitely. But, in short-lived species, there's often this trade-off, perhaps favoring rapid, immediate responses like immunity over long-term maintenance, like perfect DNA stability.
Okay.
So the default state in humans and mice is where this protein actually inhibits the HR repair mechanism we need to stabilize the genome and ultimately delay aging.
It's like having a crucial maintenance worker who, instead of fixing the problem, actively blocks the real repair crew from even getting to the site.
So that's a pretty good analogy, yeah.
Okay, that sets the stage perfectly for the naked mole rat. If human cGAS is the saboteur, the central discovery in this deep dive is that the naked mole rat cGAS has completely flipped the script.
Exactly. It doesn't suppress HR repair. It significantly enhances its efficiency, boosts it.
Wow. And how did it manage that? A whole new system.
Well, what's truly revolutionary here is the evolutionary economy of this change. This functional reversal isn't because of some entirely new, complex protein system. It's mediated by the substitution of just 4 specific amino acid residues.
Just 4. Out of the whole protein.
Just 4. Within the cGAS C-terminal domain, specifically S463, E511, Y527, and T530.
4 tiny changes. And suddenly, you potentially shift from a short-lived rodent to one pushing 40 years. That seems incredibly lucky, evolutionarily speaking.
It's remarkable.
Is there any precedent for cGAS being repurposed like this elsewhere?
Not really to this extent, no. That's why this is such a landmark finding. It suggests that maybe this specific cGAS function-regulating DNA repair is actually an ancient, conserved one.
Uh,
And the naked mole rat simply optimized it for survival and longevity, while humans and mice perhaps optimized it differently, maybe prioritizing that immediate immune defense.
okay. But it still begs the question, how? How do 4 specific amino acids dictate whether a protein drives aging or drives longevity?
Right. Let's dig into the mechanism, because this is where the molecular choreography gets really interesting.
Yeah, what are those 4 amino acids actually doing to change the outcome of DNA repair so dramatically?
Okay, so the difference really boils down to how quickly cGAS is removed from the site of the DNA damage. In human and mouse cGAS, when DNA damage occurs, the protein gets tagged almost immediately.
Tagged how?
Through a process called ubiquitination, specifically, K48-linked ubiquitination. You can think of ubiquitination here as applying a sort of molecular get-out-now tag.
Exit tag. Okay.
Exactly. And this tag then signals a protein complex, which includes something called the cGAS P97. P97 acts like the extraction crew.
The bouncer.
Yeah, the bouncer is good. P97 grabs the tag cGAS and immediately rips it away from the chromatin, the DNA complex, where the damage actually is.
So in TGAS spots trouble, maybe causes some, gets tagged, and is immediately dragged off the scene by P97, and its rapid removal messes up the HR repair process.
Precisely. It disrupts that delicate process.
Yeah.
But in the naked mole rat cGAS, those 4 evolutionary changes, the 4 amino acid swaps, significantly weaken the ability of the tagging enzymes, like one called 2RM41, to apply that K48-linked ubiquitination tag.
So it avoids the tag.
And critically, it also drastically reduces the interaction with P97, the extraction crew.
Ah, okay. So the NMR cGAS avoids the exit tag and also doesn't interact much with the bouncer. So it stays put.
Exactly. Instead of rapid dissociation, the NMR cGAS maintains prolonged retention on the chromatin right there after the DNA damage event.
And what does it do while it's hanging around?
By maintaining that prolonged presence, it transforms from a liability, like in humans, into a highly effective scaffolding platform.
A scaffolding platform, meaning it helps organize the actual repair machinery.
Yes, precisely. Its stable presence facilitates the physical connection, brings together 2 crucial repair factors. One is RA50, which is a canonical, well-known factor essential for HR repair. The other is FANCI, a factor traditionally associated with a different repair pathway, the Fanconi anemia pathway.
Interesting. So it's bringing together players from different teams almost.
You could say that. By bringing FNCI and RAD50 together, the NMR-cGAS acts as a crucial molecular matchmaker right there on the damage site.
And that resulting FANCI RAD50 complex must be the key to the big enhancement in repair.
That's the idea. This scaffolding action dramatically boosts the recruitment of RAD50 to the damage site, thereby potentiating or enhancing the overall efficiency of HR repair.
Got it.
And here's a really crucial point. This entire process, this genome stabilization boost, seems entirely independent of cGAS's canonical immune-related enzymatic activity. The mole rat literally repurposed the protein's structure for longevity.
That is just profound work to show at the cellular level. But the researchers didn't stop there, did they?
No, they didn't.
Because the ultimate question for us, really, is can you take this evolutionary cheat code driven by just 4 amino acids and use it to actually confer anti-aging benefits across different species?
And they tested this across the aging spectrum, starting small. At the cellular level, simply overexpressing the naked mole rat cGAS effectively suppressed stress-induced cellular senescence.
That's when cells just stop dividing because of damage, a key feature of aging.
Exactly. And if they mutated those 4 key amino acids back to the human-like sequence, that protective effect vanished completely.
Wow. And human cGAS.
Conversely, introducing human cGAS actually promoted senescence in those experiments.
So it really is like a clear binary switch controlled by those 4 residues.
Mm-hmm.
Okay, what about whole organisms? Did they try it in something like fruit flies?
They did, and the results were pretty striking. In Drosophila, overexpression of the NMR cGAS extended both the mean and the maximum lifespan.
Longer life, okay. But did it improve health span, like how they age?
Yes, and importantly, it mitigated several visible age-related dysfunctions. For instance, aging flies get clumsy, their motor coordination declines, which you can measure with a climbing assay.
Right.
The NMR-cGAS mitigated that motor decline. It also decreased their age-related vulnerability to pathogens and reduced abnormal cell proliferation in the gut, which is another common feature of aging in flies.
That's fascinating, taking this molecular fix from a weird rodent and seeing observable whole organism improvements in health, movement, resilience in a completely different creature like a fly. But what about mammals? Did they go there? That's the real translational test, isn't it?
It is, and this is where it gets potentially very relevant for human health. They delivered the NMR-cGAS, specifically engineered to be the best scaffolding molecule, not the immune signaler, into aged mice. These were 17-month-old mice, which is quite old for a mouse.
How did they deliver it?
They used a safe viral delivery method, an AAV, and then observed them over the next couple of months.
Okay, moment of truth. What visible anti-aging effects did they actually record in these old mice?
Well, the improvements were tangible and directly tied to established hallmarks of aging. The treated mice showed a significant reduction in their overall frailty index.
That's a comprehensive measure of health, strength, mobility, right?
Correct. And they had noticeably attenuated visible signs of aging too, like reduced hair graying and fewer age-related skin lesions.
Wait, reduced hair graying? Seriously?
Yeah.
From this one protein tweak?
That's what the data showed. Furthermore, markers of systemic inflammation in their blood, things like IgG and IL-6, which typically spike with age, were significantly lowered.
So you're telling me that this specific molecular component, essentially designed by 4 amino acid changes in a mole rat, can apparently reverse visible signs of aging, like hair graying, and reduce frailty in an entirely different mammal species.
That's what this study suggests, yes. It's a pretty powerful cross-species demonstration that optimizing genomic stability, even through tweaking a negative regulator, can potentially roll back the clock on aspects of tissue aging.
And crucially, was this dependent on those 4 amino acids?
Absolutely critical point. They showed that all of these benefits, the reduced DNA breaks, DSBs, fewer senescent cells in major organs like the liver, kidney, and intestine, reduced frailty, were entirely dependent on those 4 specific amino acid residues in the NMR-cGAS.
And if they used the human version?
If they injected the mice with the humanized cGAS instead, or the NMR version with those 4 sites mutated back, the benefits disappeared completely.
Incredible. So what does this all mean then, wrapping up? The naked mole rat, the longest-lived rodent we know, didn't just evolve stronger versions of genes we already knew were positive. It pulled off this amazing trick.
It evolved 4 incredibly precise, tiny changes in a normally negative regulator cGAS to turn it into a positive structural force for genome stabilization.
And this single adaptation seems to successfully counteract cellular senescence, delay organ aging, and extend health span, not just in mole rats, but potentially across vastly different species, including mammals.
It really makes you think, doesn't it? It raises an important question for future research and maybe even therapy development. You know, historically, anti-aging interventions have often focused on amplifying known positive regulators of things like DNA repair boosting the good guys, like SERT proteins, perhaps.
Sure, it makes sense.
But this deep dive demonstrates the potentially massive power of optimizing a negative regulator, flipping its function.
Turning a foe into a friend, basically.
Exactly. It suggests a potentially new, highly targeted intervention strategy for human aging. Maybe we should be looking for and finding ways to flip other existing negative regulatory switches in our own genome. Perhaps we can unlock latent repair capabilities we already possess and promote genuine longevity and health span that way.
A truly paradigm-shifting perspective. Instead of just hitting the accelerator, maybe we need to find the brakes that are stuck on and release them. Fantastic stuff. Thank you for walking us through that molecular marvel.
My pleasure. It's a fascinating research.
That concludes our deep dive into the naked mole rat's secret to longevity and the surprising power of just 4 amino acids. Don't forget to like and subscribe to the Sans Science Experience and share this analysis with anyone you think would enjoy a shortcut to being well-informed. Join us next time for another deep exploration into the latest science.